Hello, everyone. Thank you for joining this installment of Boyce Thompson Institute's new Breaking Ground discussion series. My name is AJ Bushy. I'm in the communications department here at BTI. And for this month's Breaking Ground, we're welcoming our first guest from outside the Institute, from outside BTI, Karen-Beth Scholthof, Professor of Plant Virology at Texas A&M University. And Dr. Scholthof is also a science historian with a particular interest in the history of BTI and plant science research. Hi, Karen.
Morning, AJ.
Good morning. And we're also joined by Paul Debbie, BTI Research and New Development. Hey, Paul. And so today's discussion is going to be a real treat. We're going to be going back in time to talk about what is arguably the birth of virology, and not just plant virology, but the entire field in general. And we'll hit some other highlights from BTI researchers over the years, including some neat stuff that's happening inside our labs today. And we chose this topic because obviously, like, viruses are on just about everybody's mind right now, particularly the coronavirus that's causing the COVID-19 pandemic. And a lot of people don't know, I know I didn't know, that virology started with plant science, and it started at BTI. I didn't know that until Paul told me, shared that with me a few months ago. So we thought this would be a really neat topic to talk about. And so that's what we'll be doing today. And a couple quick housekeeping announcements. During the session, all participants will be muted, and you can ask questions. There's a chat box. There's also a Q&A box. You can put your questions in either of those, and we'll do a Q&A session at the end of the discussion. And this, the whole thing goes an hour, so we'll be done by 1. And normally I try to keep it like a half-hour discussion, half-hour Q&A. I suspect this discussion may go a little longer, but we'll still try to keep a good 20 to 25 minutes for a Q&A. And all right, let's get going. So Karen, we'll start with you.
Okay.
Could you tell us a little bit about yourself and your research, what you do down there in Texas?
Yeah, I've been at Texas A&M for a little over 25 years now, and am a professor in the Department of Plant Pathology and Microbiology. I came up through land grant systems, training in botany, and then getting into plant pathology as an undergraduate, and trained at University of Nebraska with Myron Brackey doing my master's, and then I went to University of Kentucky and worked with Bob Shepard for my PhD, and then to Berkeley to postdoc with Andy Jackson. So I've been doing virology for a while. It wasn't until around 1997 that I really started getting interested in the history of tobacco mosaic virus. And since then, I've been primarily working on the work of Francis Holmes through archival history research. And most of this work has been funded through NSF. And it's been a lot of fun doing in parallel research in my lab and looking back at earlier work.
So you've been here before, right?
Yes, I was on.
I'm actually in.
Yeah, I was on sabbatical 2009 at Cornell in the science STS program, and so yeah, I'm familiar with BTI and the fantastic facilities that you have.
Well, thank you so much for your interest in BTI, and I'm glad I've talked to you before. You have a wealth of information about the early history, the early days here. So we appreciate you joining us today. Our other guest, Paul Debbie, could you tell us a little bit about yourself and your role at BTI?
Yeah, sure. Thanks, AJ. Like you said, my current position here at BTI, I'm the Director of Research, but I'm also what we call the Director of New Business Development here at BTI, and I'll explain what that means in a second. Just to give you sort of my history, I've been at BTI for 20 years now, starting in January. I've held several different types of positions here at the Institute. Prior to coming to BTI, I actually was a graduate student at the University of Paris in Miroslav Radman's lab, where we studied DNA repair. And I actually left graduate school to start a biotech company in Colorado, with Miro and with another scientist that I worked with. So I was out in Fort Collins, Colorado, and then in Denver for 9 years, working with that particular company. While I was doing that, I also went to business school and got an MBA. And then when I came back here to the East Coast, I was actually hired by Greg Martin, who's joining us here today, in a technical position actually to set up and run a gene expression profiling facility using microarrays to study tomato gene expression profiling back when microarrays were all the rad new thing. And so I did that for probably almost 10 years, really, in a sense. But at the same time, I also transitioned to doing tech transfer work at BTI. I managed our intellectual property portfolio and did licensing, things like that for the Institute as well. And that morphed into the position I have now in new business development, where we try to take technologies out of the Institute and start new businesses with them. And also, like I said, I also have this other hat that I wear, which is the director of research here at BTI. And like many scientists, when I was a kid, I read the book Microbe Hunters that was written by Paul de Kruif and got me really excited about science. But it also got me really interested in the kind of thing that Karen works on, which is the history of science and where it comes from. So that's, I think, one of the reasons why I'm joining us for this topic today. So that's pretty much my story. The other thing I'd like to do just before we get started is I also want to welcome the other emeritus faculty that we have here joining us this morning too, because it's pretty cool. Many of them were actually at BTI in Yonkers before we moved here to Cornell. So they really share a big part of the history of BTI. So welcome to you guys.
All right, thank you, Paul. And so the first question is for you, Karen. So you mentioned Francis O. Holmes and your particular interest in the tobacco mosaic virus, or TMV. Could you tell us a little bit about Holmes and what he did, what his impacts have been on the field of virology?
Yeah, it's so coming up as a plant virologist, you've learned some classic techniques, purifying viruses, inoculating plants. And when I was coming up into plant virology in the early 80s, we still learned how to do host range tests, which would be to take a virus and rub it onto a lot of different plants, see which ones were susceptible, and also use it to figure out the best plant to purify virus from, in terms of the virus would accumulate in the plant and not kill it. This work and knowing about resistance, gene for gene resistance, all of this stuff that we consider common or obvious now in plant pathology and plant virology was really predicated on the early work of Francis Holmes. He did his undergraduate work at MIT, worked a little bit on fungi, and then he went to Johns Hopkins to get his PhD and was trained as a protozoologist. He was then hired by L.O. Kunkel, who was the director of BTI when it first started. And his job was to find what this organism or thing or perturbation was in the tobacco mosaic virus with infected plants. And there was still a lot of controversy up until really the early 1920s about what is a virus. Was it a physiological problem? Was it an enzyme? Was it a novel agent? And so Holmes really came in and was the first, in a sense, modern scientist to really work on the virus problem. And he stayed at BTI until around 1929, 1930, when Kunkel was invited to bring the virology group to Rockefeller Institute for Medical Research that had a essentially an agricultural campus in Princeton, New Jersey that did animal and plant diseases. So at that point, Holmes moved on to Rockefeller, and he stayed there for the rest of his career. But his early work at BTI is, it remains important today, actually. And if you want me to go into a bit more detail, I'm happy to do that.
Yeah, sure. Yeah, this local lesion assay, you know, people still use it today. You mentioned, you know, you used it. I seem to recall using it in my plant physiology lab many years ago.
Okay.
So could you tell us a bit more about that, like how he came up with it?
Yeah, so this was his search for the virus agent, and he started out using something called ultramicroscopy, so essentially light microscopy, and trying to find this infectious agent in the plant. And he would have to dilute the sap from infected plants. And he spent 3 years trying to see the virus. So for those of you that are in the lab today, don't get too discouraged. Sometimes it just takes a while. And he eventually thought, well, I wonder if there's even any material, infectious material in these sap samples that I'm putting under the microscope. And so he started inoculating plants with the sap extracts. And he, as I show you in this first slide where it says 500 plants, this is initially how he set it up. And of course, BTI then and today have fantastic facilities. So he would grow up these flats of hundreds of plants and inoculate them, rub some sap on them, and then wait and see if they looked infected, if they had a mosaic or mottle discoloration. And then he could count that, yes, the sap extract did have some infectious agent, even though he couldn't see it. In time, he started noticing that some of these plants had small chlorotic halos. And he then realized that, this is actually evidence that there is an early stage of infection. And he then went on to explore a number of different plants, as shown on the figure on the right. He then was able to make a little device with insect pins and essentially a pot label or tongue depressor, and take the sap and poke holes in the plant, which in this case shows 500 pinpricks, which would be, each pinprick would be equivalent to inoculating a single plant. So he had developed this rapid biological assay now. And from that, he then came to the realization that when there were local lesions, as shown on the bottom figure, which is really one of the classic figures in plant pathology, of his Nicotiana glutinosa assay, that it would develop small necrotic dark brown lesions within a couple of days. And so he could essentially titer the virus, still not see it in terms of way we would think with electron microscopy today, but he could see that there were lesions, and they were you could relate them back to the titer in a sap extract.
So just to clarify real quick, so all those 500 pinprick, like one pinprick is coming from that has the sap of one plant on it?
It has, now you can grind up a plant, and you see the efficiency isn't super great, but efficiency of inoculation isn't necessarily great either. So from either the dark lesions or these chlorotic halos, that would indicate an infectious unit from a plant. So if you're familiar with plating bacteria or doing phage type assays, this is a type of quantitative, maybe more to the qualitative side assay to determine that a plant did indeed have virus in it.
Okay. That's pretty incredible that you could test 500 plants on a single leaf. So he could do many thousands of plant diagnostics on another single plant.
Right, and in time he selected Nicotiana glutinosa because the plant is fairly short stature, and he could do. You could actually rub a leaf, and each leaf could have a different extract from different plants, because each infection would form this local necrotic lesion, which you see as these dark little spots on the leaf. And then in time, he made this remarkable observation and sort of thought experiment that that lesion represented a specific genetic interaction between the virus and the plant that was associated with resistance. So essentially a gene for gene interaction that way predated the work of Floor, who is known for gene interaction with his work on flax. And
so how exactly did he know that these were viruses and not something that you mentioned, enzymes, bacteria, everything else?
So yeah, so if you would take a healthy plant, one that didn't have symptoms, and inoculate a lot of other plants, you would not develop any symptoms. So it was not something in the plant per se, and that when you would inoculate these plants, there had been some previous work showing that it was not physiological or not an enzyme that was done by H.A. Allard of the USDA. But this very specific response of being able to do dilutions and see these lesions was really good evidence that it was something small, which we knew from earlier work of Beyerink. You could not see it in the microscope, and yet it is infectious.
Gotcha. And then later on, he did find a resistance gene, right? That's this N gene?
Yeah, so the N gene was for the necrotic or N response. And from that, he thought, well, if it's in this Nicotiana glutinosa, maybe I could move this gene using classical genetics to a crop plant, tobacco. And the intention that he had, by then he was in New Jersey, and there's a lot of vegetable crop, big truck crop industry, so fresh vegetable market. And Holmes saw that there was a lot of tobacco mosaic virus in the tomato and pepper fields. So his idea was to make tobacco resistant to tobacco mosaic virus, so that you just have maybe a couple of these little infections and not a whole plant, obviously coming down with florid symptoms of infection and having a lot of virus. And the idea was that the farm workers would primarily be rolling cigarettes from tobacco, and the tobacco mosaic virus would get on their fingers. And then when they would do the transplants of the peppers and tomatoes, they would infect all the plants. So his initial intent really was to make tobacco resistant to TMV to protect tomato and pepper crops. And again, he got caught up in some genetic difficulties of moving this gene from Nicotiana glutinosa to tobacco, which entailed another 3 years of frustration. And he was actually first successful in moving the resistance gene, the L gene from pepper, also to tobacco mosaic virus, from Tabasco pepper to bell pepper, and showing that resistance, genetic resistance, could be moved across species and be used out in the field.
It's very cool. I know we could keep talking about him for quite a bit longer.
It's amazing.
Yeah.
I just have to say, it's really very thoughtful work, very minimal tools available.
and that was really the birth of virology. Is the first one who said, okay, here's the virus. Like you said, you couldn't actually see it like under a microscope, but you knew that there was one at that site of the pinprick.
Yep.
That's pretty amazing.
Yeah.
What's interesting about that kind of discovery, work is that it's like they knew what it wasn't. It's like,
Yes.
we know it's not a bacteria, but we don't know exactly what it is. And it's a really cool overlap with applied research at the same time. I mean, they were trying to solve a real problem for tobacco farmers.
Yeah, and I think that's a really nice lesson for today is, this from bench to lab and back again, translational research really in both directions. And, from his work, there, became enormous amount of work done on putting resistance genes into crop plants. And then on the other side, the early biochemists, people like Wendell Stanley, who went on to win the Nobel Prize for purifying tobacco mosaic virus, that they used Holmes assay to determine if they had any virus when they were doing these huge purifications to be able to crystallize the virus and then actually see it in a more contemporary sense. So it was, yes, he was important in both directions and well respected in both venues of applied and basic research.
Real quick, we did get one quick question that I'll, since you just mentioned moving the Tabasco L gene into bell pepper, was that the first example of a resistance gene being moved between species? Do you know the answer to that question?
So people had been breeding for resistance mostly by selecting plants that were resistant in the field already. So far as I know, this is really the first deliberate use of genetics to move a single resistance gene to another plant.
Gotcha, makes sense. Okay, let's move on to our next cool scientist, Helen Purdy Beale. And she was a contemporary of Holmes, correct?
Yes. Yes.
Back in the 20s. I think she was here until like the 50s maybe.
Yeah.
Could you tell us a bit about her and her contributions?
Yeah, so Beale is tremendous, and in many ways, I think forgotten. That and in some way, Holmes's work is remembered, but in some ways forgotten. Well, it's not a hypothesis. My notion of this is that none of them had students because they worked at private institutes, and so there weren't necessarily students to carry on their legacy, but their methods have been brought across through the years. Beale was, she went to Barnard in New York City and then completed her PhD at Columbia, and was hired on early on at BTI, part of the first wave, and in this virology group. She had also worked at the with the sugar Hawaiian sugar planters group. I'm not getting the name quite right, but she had been in Hawaii. Kunkel had also worked a bit in Hawaii. And she became very interested early on in using serology to detect plant viruses. And while at BTI, she developed these assays where, as shown in this little cartoon here, that you could take sap from a healthy plant and inject it into a rabbit. The purpose is to make antibodies, and also take sap from a tobacco mosaic virus infected plant, and through precipitation assays, demonstrate that there was something different in the infected plant Okay, so this is following along with Holmes of trying to figure out what is this virus agent. And you could use this SAP serum to actually use the antibodies to bind up either healthy proteins or the tobacco mosaic virus protein as part of the virus particle, and show that there was indeed this infectious agent that was in the infected plant. Her experiments, I think, needed a bit more. If you want to understand them, you need to get a bit more into serology and understanding the notion of these. But again, these are really logic puzzles, much like Holmes did. And today they seem very straightforward, but you have to realize at the time, people didn't know what antibodies were, people didn't know what viruses were, but they knew that you could precipitate out proteins or proteinaceous substances. So she did these assays. She was a tremendous statistician and developed these very complicated assays to determine what was going on in the plants, and then working with the serology. And today serology is, I mean, it's important for everything from worrying about a COVID assay to a grower or plant disease clinic sending samples out for a quick and dirty analysis, and even, you know, little strip tests that you can do just in the field to see if you've got a virus infection or other problems.
Yeah, so I was going to say, like, when
in the world to work on these virus issues.
And this work also helped you help researchers be able to track and trace plant viruses, right? Confirm different strains from each other because the antibodies were so.
Yeah, and so you know, as time went on, things became more sophisticated, and you would be able to determine very minor differences. Say for tobacco mosaic virus, the major protein, or the outer coat protein, may have small differences in amino acids. And with serology, you can show this, and with, again, different types of precipitation assays, you would be able to detect different strains of viruses. So as the early biochemists came in, and they wanted to, and we're doing mutagenesis, chemical mutagenesis, UV mutagenesis, that if there were changes in amino acids, before sequencing was possible, you could use serology to show there had been changes.
Yeah, this is all, you know, just to kind of bring it back to our present day. You know, this is all so important today. You know, when I think of serology, I'll be honest, I never thought of it in terms of with plants. I just thought of it in terms of health. You know, and you know, I gave blood a couple months ago, and the Red Cross, they test your blood to see if you have any antibodies against the COVID coronavirus. Like, that's serology. And that, you know, and she was doing that in plants back in the 1929, I think.
Yeah. It's very amazing. And you know, if you have the ability to continue, you know, to work and develop antibodies, that, you know, animals for many things are still quite important for research. Moved away from that a bit, doing you can clone antibodies. I mean, there's other things, but there's still it still is really a framework for a lot of the work, whether you're working on green fluorescent protein in plant cells or animal cells and you want to detect them, or confocal microscopy. I mean, there's all kind of instances of using antibodies, as well as you know, in the lab for Western blots and all sorts of other things.
Okay, so I'm keeping an eye on the time. I'd like to move on real quick. Our third person, and that is George McNew. He developed this thing called the disease triangle, which I have seen, and you know, related to the current pandemic. Could you tell us a little bit about that and why it's relevant today?
Yeah, so I'm sure that several of you that are participating today probably knew George McNew. He was the director of BTI for a time. So it's funny, I don't know that much about McNew because I haven't written anything about him yet, but I've written on the disease triangle and its utility for plant pathology and also for public health. Any student who has taken Introduction to Plant Pathology sees the disease triangle. Generally, on the picture that's on the bottom left as indicating some interaction occurs between a host, a pathogen, and the environment that determines the incidence of disease. These two drawings that I've made here are, to show then why in time of COVID, this is important, because if you change the environment, hand washing, masks, social distancing, you'll have less disease because you're not allowing the virus to move around as much. This very simplified diagram that is occasionally used for public health, sometimes as a disease triad, came from the upper drawing that McNew presented around 1960. And it is on the surface of it is very simple and yet extremely complex, that it really details the parameters associated with modeling and forecasting disease incidence and outbreaks, and a clear attention also to time in duration of conditions that are favorable to the pathogen, the age of the plants, the prevalence of the pathogen. It's very specific environmental conditions. And so this is, I just think, one of the most amazing insights that we've had in plant pathology, and still very useful today. Even though we've gotten away from it, I think very rarely do people see his original triangle, but you mostly see the simplified version. But it's, we have disease forecasting. I mean, Cornell is renowned for its disease forecasting work for late blight. There's forecasting for apple diseases. So I think it deserves more attention. That's what I would say about the disease triangle. And again, I just want to say it is truly remarkable how many important scientists have come out of BTI. And I'm just looking at those in the past. The legacy of these people is shown by the work that's going on today.
Yes, and that's a good segue into my question for Paul, and that is, stuff that's been happening more recently at BTI in the field of virology, not just plant virology. I purposely, the title of this is not the history of plant virology at BTI, it's the history of virology at BTI, because there's more than just plant virology done here. And could you talk a little bit about some of the more recent things that have happened here, Paul, and some of the stuff that's still going on today?
Yeah, sure. Go ahead, bring up those slides that you had. Helpful. Yeah, so why don't I tell you a little bit of everybody about a little bit of the story of the recent sort of work that's gone on in BTI. And when I say recent, I mean, it's a number of decades, let's say, of work at BTI. And some of it spreads from the work that was done in Yonkers and moved here to BTI. One of the people who's joining us on the call today is in the middle of this picture here between myself and Dan Klessig. That's Bob Granados, who worked with this little bug on the right here, which is the cabbage loper. So, and one of the cell lines that Bob developed actually comes from this particular insect, and it's called the high five cell line. So this was in the late 80s when Bob was doing this work at BTI. And it's very interesting. You know, we've got Karen here from Texas A&M. One of the reasons they were using these particular cell lines was actually to do what we call baculovirus expression work with these insect cell lines. So Max Summers, who was at Texas A&M, had developed this method to actually use these naturally occurring baculoviruses, so it's an insect virus, and use it to do protein expression work. And Gary Blissard here, who's currently at BTI, continues.
So real quick,
Sure.
Paul, when you say expression, you mean like putting a gene for a protein inside the baculovirus and then?
Yeah, so we have kind of a mixed audience here. Why don't I just briefly kind of explain how it works, if that would be helpful.
That'd be good.
For people.
Yeah.
So yeah, so basically you take your gene of interest and you put it into the baculovirus. You engineer it into the baculovirus, and then you make a bunch of that virus that has your gene of interest, and you then take these insect cell lines and you infect them with that engineered or recombinant virus, as we call it. And then you then allow the machinery of the insect cell to generate that protein that you're interested in. And then you then take either the extract from that cell line or what's been secreted by the cell line, and you purify your protein of interest from it. It's a two-step process in most cases. Some of the newer engineered cell lines, for example, that Gary is working on would make it a single-step process where you actually engineer the cell line to have that gene of interest in there, so you could do it as a one-step process. But normally it's a two-step process the way we do it now. And of course, one of the highlights that came out of this particular work was we licensed this cell line to a company called MedImmune back in around 1990 or so, and they eventually became it eventually got to the point where GlaxoSmithKline actually made a human papillomavirus vaccine, an HPV vaccine. There's a couple of different ones on the market now, people have probably heard of. And so one of those is actually made in this high-5 cell line that came from Bob's lab here at BTI.
That's very cool.
Yeah. You know, the other thing I would mention, you know, because it's kind of interesting and somewhat timely, someone sent me an email just before this meeting, actually, where it looks like we found another COVID-19 vaccine candidate potentially that's being made with the high-5 cell line.
That's great.
There's a couple of different groups that are doing that work right now. You talked about, and Karen was talking about serology work early. Some of the proteins that are needed for serological work with COVID-19 can be made in insect cell lines, including the high-5 cell line. For example, you need to make the spike protein. You need to make the human receptor proteins to do a lot of the serological assays. So there's a lot of utility for these cell lines just in sort of reagent grade work around the COVID-19.
It's very amazing to think that, you know, these cell lines from BTI, had, you know, originally out of Bob's lab, could be used for a COVID vaccine and save lives, and, you know, and these serological tests to help, you know, to help keep surveillance and keep it in check. That's pretty neat.
And just to tie it to our current work, we have a group that we collaborate with in Austria who's currently has what we call the second generation of high-5 cells. So these are new and improved versions of the original cell line that Bob discovered. And Gary also was involved in this particular work. And so we have ongoing work with our collaborators in Austria and others that are actually using the second generation of high-5 cell lines to do some of this work, including that group is actually working on a COVID-19 vaccine in that particular cell line.
Very cool. All right, well, we're at about 12:45, so I want to thank you both. That was an awesome discussion. I feel like we barely scratched the surface of many different topics. So I think we could go to Q&A. I see one question in there, and if anybody else has any questions, you can type it in the chat or the Q&A box. So this looks like it's for Karen. Would you care to comment on the relationship of Holmes's work to Beijerinck, maybe, in Holland, and with Bodden and Peary in the UK? sure I butchered their names. I apologize.
So it's a pleasure to take a question from David Balcom, and a treat. So Martinez Beierink was a virologist, well, no, not a virologist, what am I saying? A microbiologist in Delft in the Netherlands. This is sort of a hotbed for microbiology. Leeuwenhoek, who developed the first microscope, was from Delft. Beierink, to me, was really the first one to show that tobacco mosaic disease was a virus, which he referred to it as contagium vivum fluidum. Ivanowski in Russia had done some similar experiments with filtration, but he didn't trust his results, and had a bunch of hand waving about maybe it was bacteria or something. But Beierink did very thorough studies and showed, as Paul was indicating earlier, that defined viruses as what they were not. They were not bacteria, they were not visible. So, essentially there's still these unknowns, but they were thought to be a different type of agent. So Beierink in 1898 really sort of kicked all this stuff off, and this is where we get the modern term virology. The work of Bodden and Puri goes beyond being beautiful and important. Bodden was probably one of the most brilliant scientists of the 20th century. So I said that Stanley had crystallized tobacco mosaic virus by the mid-30s using Holmes's local lesion assay, basically as a bioassay. Stanley thought he had come up through this protein work with Northrop, and he thought TMV was a protein. Bodden and Puri very quickly and strongly showed that there was actually phosphorus associated with this crystalline structure, and that it was likely, some kind of nucleic acid going to be associated with that. Stanley took some time to come to agree with them. But Bodden, I still use his plant virology textbook for my history research. That incredibly clear, brilliant thinker on RNA viruses and general science.
Mm-hmm. That's fantastic. Another question from Bob Granados. Did Holmes and Orbeel get national recognition for their research? Louis Kunkel was elected to the National Academy of Science.
This Bob, thank you for that. It is, I don't understand why Holmes was never elected to the National Academy. Kunkel was very well placed, having come into BTI and then as the director for the plant virology research at Rockefeller. I do not know why Holmes was not elected, and Beale, nothing. I mean, it was, a different time, and it's not an excuse. It was just a different time for women in science then. She stayed at BTI. As far as virology, after Kunkel took the group, with the exception of Beale, to Rockefeller, she continued to work and do very good work. She also, I just happen to have this on my shelf. She developed this bibliography of plant viruses that is about 1,500 pages long, where she went and found every single plant virology and serology paper that was known through the 50s or so. But her serology work was not recognized. I think, let's see, Holmes, of course, received all of the most esteemed awards from the American Phytopathological Society. But Beale, yeah.
She seemed really impressive. I noticed when I was reading her paper from 1929, where she describes, injecting the rabbits and getting what turned out to be antibodies. She was the only author on that. There were no co-authors. She did all that herself.
Yeah, and you'll see with Holmes too that he had no co-authors, and I've always been especially curious about if he had any technical staff and who was doing all the greenhouse work, because he could plant thousands of plants to do his experiments, and if we never have enough growth chamber space anymore, and so there had to be some characters in the background, but there's no evidence of that looking at the contributions of the Boyce-Thompson Institute, the series that was produced, or all of the papers that were published throughout the years.
Yeah, that's interesting. But there's some written stories from way back then. If we only had people who knew them. Well, that's the last question here. So I'm going to ask one more of Paul on the slide we showed with you. It was there was a picture of you and Bob Granados and Dan Klessig. Mm-hmm. And Dan is another one who started out in virology, correct? You want to talk a little bit about Dan's early work?
Yeah, it's interesting. I think as best I understand sort of the history of his science, he worked with adenoviruses early on before he switched over really to working in plants. He always kind of jokes that he made the mistake making that switch because there's so much more money in the NIH for the adenovirus kind of work than you have, or virology kind of work than you do in plants. But yeah, so I mean, it was really sort of an evolution from, the interest that he had in adenovirus work, and that evolved into getting into plants, and not necessarily a plant virologist, but a plant pathologist, I think you would really say. So I don't know if that answers your question or not.
Yeah.
The other thing I would point out, this is kind of a different, somewhat related topic, but different. We talk about virology work and the importance of, that specific kind of research. But I think one thing that's really important to think about is also, the environmental sort of work that we do at an institute like BTI, where we're studying plants and how they interact with the environment. Because if you look at that disease triangle, one of the things you have there is that favorable or unfavorable environment for transmission of viruses. And it even gets into things like, we create a favorable environment for a coronavirus to spread from a bat into probably another mammal and then into humans. So there's a lot that we need to learn and understand about the environment and our interactions with it, and why we're causing pandemics in a sense. We know the environment's changing in a way, and that's another part that we really need to understand in the plant world, I think, and why plant research is so important in a field like virology.
And it was the original premise of Boyce-Thompson as he developed the Institute. It was the Institute for Plant Research. And so I'm only familiar with the virology aspects, but there were many other aspects of getting into plant improvement, breeding, chemicals, early work on both pesticides, insecticides, plant growth regulation. So yeah, what Paul says is, it's incredible that the intent has been maintained for all these years, that you're looking at more than 100 years now since Thompson first, thought to develop the Institute.
Yeah, we opened in 1924, I believe. So we're coming up on 100 years since it opened its doors.
I think that's really, one of the things we're trying to do here today, right, is, plant, research has really not gotten its fair shake, let's call it. Like when you hear Anthony Fauci talk about the disease triangle, they don't even mention the fact that it came from plant work, you know. So it's kind of our job to make sure people understand how important the work is, and because sometimes it kind of gets lost in the translation, I think.
Right.
All right. Well, I think we're there. Thank you so much to you both, Karen and Paul, for sharing so much about plant virology. I want to thank everyone out there who joined us for this chat, for this discussion. It was very good. We had some great questions in there. And you can read about some of BTI's current plant virus research and many other neat stories about BTI science in our 2019 annual report, which you can find online at btiscience.org/annual report. And I see Mike pasted that URL in the little chat box. And the 2019 annual report is all digital this time around with 3 different views. There's a single page view, a 2-page view, which is most similar to seeing like the hard copy if you had it in your hands, like a PDF, and a reading view, which is like reading any story on the web. There are many links to videos and other neat interactive aspects in the online annual report. And I want to invite everyone here to next month's Breaking Ground discussion series, which will be with BTI's Fei Wei Li on Thursday, October 29 at 12 noon Eastern. And we'll be talking about spooky plants in advance of Halloween. And if any of you have never seen Fei Wei speak before, you should definitely join. She's always a treat. In this case, maybe trick or treat, I don't know. And so it's going to be a fun one. And you can go to btiscience.org for more information and to register. And yeah, thanks again for all your support. BTI is an independent nonprofit research institute, and we operate in part thanks to the generosity of community members like you. And if you'd like to make a gift to support BTI, you can donate online at btiscience.org/give, or you can email our development team at development@btiscience.org. And again, thank you all for your interest in your interest of the Boyce Thompson Institute. And I hope you have a wonderful rest of your day and going into a weekend. All right, have a wonderful day.
Bye, everybody. Take care.
Thanks, AJ.
Yeah. Thanks again, Karen, for doing this with us. This is great.
It's great to talk about BTI.
We'll have to have you on again sometime.
And yes, anytime. Thank you.
All right. Take care, everybody.
So long.
Bye.